So welcome back for one more video in quantitative genetics chapter. So here my flow of lecturers again will differentiate or know about continuous and discontinuous variation, and then we'll see what are a few examples in continuous variation, and in both continuous and discontinuous, which is called as quantitative characters estimation or mostly dealt in quantitative genetics. And then we'll have an introduction about genetic and environment interaction, then different models which are used in genetic and environment interaction, and what is the significance of this genetics and environment interaction with respect to disease, and then followed by the references which I have used for making this PPT. So first of all, lot of studies in genetics happen with Mendelian characters, what we say. Mendel used pea plants and he did not use the herb or the weed which is present in the garden, but he selected pea plants and which has a clear-cut segregation and clear-cut distinct or separate phenotypes which he was able to quantitate. That is how he came up with his laws. So discontinuous characters is found in population in two or more forms, with as they are very distinct and separate forms called the phenotypes. The two phenotypes can be easily distinguished, or they can appear as polymorphisms also. So if you have mutants, where more common normal phenotype will be called as the wild type and the mutant will be called as a mutant. Examples of discontinuous variations are the blood groups, the skin pigmentation, the albino occurrence, or the tall and short genotypes of pea plant. So when few more example is the seed is winged and non-winged, the fruit. That is very clear whether the fruit is winged or non-winged, which helps in dispersal. But beside that, there is a small picture which tells about variation in height, branch number, and flower number in a herb. So you can see that there is no clear differentiation between two characters, like for example height. If you take, there is no clear differences like tall and short, and there is a gradation or variation from the highest to the lowest. So a character showing continuous variation has an unbroken range of phenotypes in a population. Like for examples of continuous variations are height, weight, skin, hair color. So in between phenotypes you can find. But in case of discontinuous variation, you find the extreme phenotypes. In case of continuous variation, along with the extreme phenotypes, you find the in-between phenotypes also. In some, all of the variation is environmental and has no genetic basis, as is the case of different languages spoken by different human groups. In other cases, such as shades of human eye color, and the differences are caused by allelic variation in one or more genes. For most continuously variable characters, both genetic and environmental variation contribute to differences in the phenotype. So a continuous variation, there is no one-to-one correspondence. You cannot say, okay, this gene, like for in discontinuous variation, one gene is always a dominant and represents the tall character, and another genes represent a short character. Here you do not have a single gene contributing to one particular phenotype. So for example, there is one more plant here. That is Indian paintbrush. In this photograph, it shows in the left, it is very clear that it shows a range of colors, which you can see the in-between colors also between yellow and the red. So if you take two population, like for example, the petal length in case of This is an example of continuous variation. If you have two population, the length of the petals is taken, as in this plant Nicotiana longiflora. Even if you say the length of the petal is height of say 65 close to 70, but there is variation among those genotypes. If the length of the petals is 52 centimeters, then also there is variation in the parental population. So if that population is inbred, then you get the F1 population, which is an average as expected of the parental population, but in that also you see variation. If they are further bred and then produced F2 generation, the variation, the average mean reduces, but the variation increases. That can be clearly seen in the F3 generation, where the specific population, the extremes or in between can be used for breeding, and then you can see appearance of the parental type or the F1 generation, or you can see the appearance of various other intermediate behavior, intermediate lengths of the petal. That means the cross made between the individual, suddenly the offspring shows up different behavior. This behavior is a rule in most species. Most species does not show Mendelian behavior, where Mendelian used horizontal varieties, single alleles, but most of the characters vary continuously. The variation is the result of some genetic mechanism different from those that apply to the Mendelian genes that he used to study in the earlier experiments. So the continuity of the phenotypes is the result of two phenomena. Genotypes does not have a single phenotypic expression, but rather norms of reaction that cover wide phenotypic range. As a result of the phenotypic difference become very blurred. There may be many segregating loci having alleles that may make a difference in the phenotype under observation. Suppose you have 5 affecting loci and 3 alleles, so the possible phenotypes what it can form will be 3 to the power of 5 different types of phenotypes, from the parental, the dominant at each alleles, to the recessive at each allele, and the intermediate. Thus many different genotypes may have the same average phenotype. Even if the 2-3 characters additive can become a dominant character, which is similar to the one of the parental types. So the in-between phenotypes can be masked. At the same time, because the environmental variation, the two individuals of the same genotype may not have the same phenotype. Though genetically they are same, they cannot behave the same because of environmental variation. So statistical methods are used for the production of this inheritance of phenotypes in the absence of knowledge of underlying genotypes, which is the basis of quantitative genetics, like observing the mean variance, or observing the variance in a population, or observing how the two characters, one character in the parents and the offsprings are correlated, or regression analysis. All this is done in quantitative genetics. So the quantitative genetics is the study of genetics of continuously varying characters. It is concerned with the answering of the following question: whether the observed variation in the character influenced by all genetic variation, is all the variation simply the result of environmental variation or developmental noise. The meaning of developmental noise, it's a concept in developmental biology in which the phenotype varies between individuals even though both the genotypes and the environmental factors are same for all of them. Okay. So that is developmental noise. Or are there alleles segregating in the population that produce some differential effect on the character? If there is genetic variation, what are the norms of the reaction of the various genotype? How important is genetic variation? So, as a source of total phenotypic variation, does many loci contribute to variation? So, these are the few questions which are answered in quantitative genetics. So continuous characters in continuous variation in a character is sometimes assumed to be necessary caused by a large number of segregating genes, and continuous variation is taken as an evidence that a character is controlled by many genes. That is multiple factor hypothesis, that a large number of genes, each with a small effect, are segregating to produce a quantitative variation. If one of the character is limited and if many segregating loci influence it, then we expect the character to show continuous variation because each allelic substitution must account for small differences in the trait. So when we are talking continuous characteristic, it can theoretically observe assume any values between the two extremes. The number of phenotypes is limited only by the ability precisely to precisely measure the phenotype. So some characteristics like meristic characters, for instance, are also measured by whole numbers. For example, litter size. The female mouse is always a whole number. A meristic character has a limited number of distinct phenotypes, but the underlying determination of characteristics may still be quantitative. These characteristics must therefore be analyzed with the same technique that we use to study quantitative characteristics. Another type of quantitative characteristic is threshold characteristics. Like we say the disease is present or absent. So that's a threshold. Upper the threshold value, definitely there is appearance of the disease. Threshold characteristics exhibit only two phenotypes. They are considered quantitative because they too are determined by multiple genetic and environmental factors. The expression of this characteristic depends on underlying susceptibility that varies continuously. When the susceptibility is larger than a threshold value, a specific trait is expressed. So we'll take one more example where we have three genotypes. One is homozygous dominant, homozygous recessive, and heterozygous dominant. So as expected, homozygous dominant plants are taller and homozygous recessive plants are shorter in population, and heterozygous are intermediate between the population. So when the environment is kept constant, the intensity, dominant has a higher intensity, say 3, in between character is intensity 2, and distribution of homozygous recessive is 1. So there is variation in the population. Total, when you combine, that black line is the total population variation. So the result is that we cannot carry out a simple Mendelian analysis to determine the genotypes of an individual. So when we consider the total genetic population, and between the two arrow mark, if you try to predict what is the genotype, then it is little confusing because there is an overlap of all the three possible genotypes. So the genetic genotype of the plant can be homozygous recessive, or some individuals in the population can be homozygous dominant or majority heterozygous dominant. So this differentiation in phenotypic differentiation can be done by imposing some environmental character, environmental variation. Suppose you impose some character like making the genotypes to grow faster. If you increase the growth rate of the genotypes, then the homozygous recessive will grow slowly, homozygous dominant will grow very fast, and then heterozygous dominant will be in between. So you can now differentiate the three genotypes to an extent very clearly. If you see the total population curve, again the black one is showing clear differentiation between the three genotypes. So the total population is a result of three separate genotypes. So environment plays an important role in exaggerating the differences between the genotypes. So a quantitative character is one which the average phenotypic difference between the genotypes are small compared with the variation between the individual genotypes. So the environment, a particular population and its genetic makeup, the influence of environment is very important. So genotypic and environmental variation, interaction is very important. So early experiment of phenotypic variations, calculations and all were done by eliminating certain character and using statistical study, and there were two major people who initiated this, followed by Arthur Jensen, who defined interaction was purely you can define by purely statistical methods. And people started to relate interaction with environment and then genetic basis as the proper outcome of phenotypic expression. So the genotype of an organism is the genetic makeup of the organisms, which can have 2 to 3 factors, like the genotype is the total gene content or the what the gene makeup is there of the organism. Apart from that, the other two being inherited epigenetic factors and non-inherited environmental factors affect the genotype. Environment, anything apart from what you're studying is environment. It can be living, non-living, it includes physical, chemical and other natural forces acting. You can have two different types of environment, a micro environment and a macro environment. A micro environment is a very small specific area in the habitat distinguished from its immediate surrounding by factors such as amount of incident light or the degree of moisture. The environment of a single plant or an organism as opposed to that of others at the same time in almost the same place is known as microenvironment. Macroenvironment is associated with variables having large and easily recognizable effect, is termed as macroenvironment and may include differences over years, location or irrigation schedule, etc. Macroenvironment can be reviewed as a collection of microenvironment whose individual effects on organisms are quite small. So having an environmental variation, you have two possibilities. You can have a predictable component of variation, you can have an unpredictable variation. Predictable environmental variation will be like what you can induce, the photo period what you give to the plant, or any fertilizer specific type of fertilizer and amount of fertilizers what you are giving, or the specific soil type can be predictable component or predictable variation. Unpredictable variation will be like includes a lot of fluctuation, like there may be sudden annual precipitation, or can be temperature variation. Slight variation can be unpredictable, and relative humidity changes, mostly coupled with agronomic practices, are variation in the environment. So the genetic makeup and environment, as always, have an interaction. uh, If you have two genotypes and one type of environment, both the genotypes will differ in their interaction or response. So a norm of reaction is a graph that shows the relationship between a gene and the environmental factor when phenotypic differences are continuous. So a phenotype, or the possible phenotype, is because of a genotype and environment interaction. So the ability of this interaction can be in terms of correlation graphs. If you have a particular trait which you are monitoring over the environment, then the trait can show the first kind of a graph where you have parallel lines. That means there is no interaction between the genotypes. Or it can show slight interaction in terms of plasticity between the genotype, the trait, and the environment changes. And the third type of graph where there is high variable in plasticity is seen. So there is a strong genotype and environmental interaction. So gene and environmental interaction are studied to get better understanding of variation phenomena in a population, especially in terms of in epidemiology, genetic environmental interaction are useful in understanding some diseases. Sometimes sensitivity to environmental risk for a disease are inherited rather than disease itself being inherited. Individuals with different genotypes are affected differently by exposure of the same environmental factors. Thus, gene environmental interaction can be showing a different phenotypes. For example, sunlight exposure, some individual who are fair will produce skin cancer, or some individual who are darker are resistant. So that is one example. And another example is the Drosophila here, what I have shown, the bristles, abdominal bristles in different homozygous genotypes. With temperature, they can vary. So there is a correlation with temperature and different genotypes. So there are two different concepts of gene environment interaction. Some people study this concept based on statistical method, and some they monitor the developmental gene and environment interaction. So when it is like the statistical or the biometric conception has its origin in research program that seeks to measure the relative population of genetic and environmental contribution to phenotypic variation within the population. So usually population genetics and behavioral genetics study statistical modes of analysis of genotype, gene and environmental interaction. Developmental interaction is not seen merely as statistical phenomena, but in any case can cause interaction of gene and environment producing in an individual's phenotype. So there are particular models of gene and environment interaction epidemiological model. There are 5 models. Model A, where the genotype increases expression of risk factor. We'll see the examples. Model B, where the genotype exaggerates or increases the effect of risk factor. The model C, where the risk factor increases the effect of a genotype. Model D, both the genotype and the risk factor are required to raise risk. In model E, the genotype and the risk factor each affect risk. Combined effect can be additive or can be non-additive. So we'll see each one example. The first example is where there is increase the level of risk factor but does not cause the disease itself. For example, phenylketonuria. It is caused by the inability or there is a mutation in the gene where the conversion of phenylalanine and tyrosine happens. If a person is having mutation in a gene, then the phenylalanine conversion does not happen, and there is accumulation of phenylalanine, and which converts into phenylpyruvic acid, which is harmful for the body. So that if you avoid the diet, then the genotype has no effect. So you can also minimize the levels of the risk factor. In the second case, where the slight as the model B says, the genotype as highest effect on the risk factor. Model B has a direct effect on disease susceptibility, which is amplified by genetic susceptibility. Xenoderma pigmentosa is it's a rare genetic disorder, and here the people will have uh, im, uh, impaired or damaged DNA repair system. When we are exposed to UV, normally there is formation of thymine dimer, which can be repaired by DNA damage repair enzyme system. But in case of xenoderma pigmentosa, is a genetic effect where the child itself can develop this disease. So you have to take particular care, otherwise at the age of 10 only the child can have skin cancer. Another model where the genetic susceptibility directly affect the disease risk. Porphyria variegata is a disease, it's a metabolic disorder characterized by deficient function of the enzyme protoporphinogen oxidase. It's a very important enzyme which converts the coproporphinogen to porphyronogen, which is important in heme biosynthesis. If the person has a defect in this oxidase enzyme because of mutation, which is estimated 1 in million, but there is an direct effect on skin, and then the person can, if they are homozygous and it occurs mostly triggered only after puberty, they can show neurological disorders also. So another model is model D, where here it differs as neither factor in this situation can affect disease risk. However, when both genetic susceptibility and risk factors are present, the risk is increased. Okay, so the enzyme glucose 6-phosphate dehydrogenase is an important enzyme in pentose phosphate pathway. The deficiency will not be so effective because glucose can also be used in other pathways. But if there is an infection, this pentose phosphate pathway produces a lot of NADPH that is used in regenerating GSH, which helps in neutralizing hydrogen peroxide, which is caused due to an infection. The environmental risk factor, if it is present, if the pool of GSH is not replenished, then there is the effects seen. So only in case of a presence of a certain factor, this risk is increased. Another model, fifth model, depicts a scenario where the environmental risk factor and genetic susceptibility can individually both influence disease risk. Alpha antitrypsin deficiency. So, it is called alpha antitrypsin because before the normal understanding the enzyme, it's a protease inhibitor. It was named differently. So this is an enzyme which is produced by our liver. The function of this enzyme, the liver produces it and then it puts out into the blood. The function of this enzyme is to go and bind to elastase enzyme. Now, where is this elastase enzyme? The lungs, especially when they are infected with some pathogen. or cigarette smoke, some particle has entered our alveoli, then our neutrophils rush in for help, and they releases elastase enzyme, which will break down the elastin and thereby clearing the infection. Now, if this elastase enzyme is produced in large quantity, if the elastase is not neutralized, then there is formation of local sepsis, and then the inflammation can happen. So the alpha-1 antitrypsin is produced, and that goes and binds to elastase, thereby preventing our lung from damage. The gene for this alpha-1 antitrypsin can be mutated. In the case when there is an infection, the mutated protein is produced in large quantities, and that protein gets accumulated in liver, thereby causing liver cirrhosis, and then chronic obstructive pulmonary disease. Now, if there is any genetic defect, it can be seen in children only by liver cirrhosis, what is shown in the picture here. So it has two gene component. The dominant is the M type, and the recessive misfolded is the Z type, where it is accumulated in the ER of the hepatocytes. The wild type, if both the contribution from both the parents have wild type, then there is a normal copy or 100%. If there is one normal and one deficient, the mutant will have 60% protection, and there is production of this alpha-1 antitrypsin. If both the parents have a mutant gene, then there is 15% production of this enzyme, and there is high deficiency and the expression. So this is one more model of study what is seen. And the other more methods of analysis is by doing traditional genetic designs like twin studies. Twins are monozygotic twins, are twins which are born with the same genetic makeup. Suppose if they are given two different environment, here say two females where they are born with genes that absorb fat twice as fast as normal average person. If they are given two different environment, one giving a healthy food, other junk food, unhealthy food, then you have obesity genes triggered in the person who is eating unhealthy food. Then we can compare both the twins and say, okay, this is a completely environmental effect, and then understand what is the effect of the environment on genetics. Another is adoptive studies. Adoptive studies means you have a biological parents which have certain characteristic, and the child is adopted with the adoptive parents. So both the parents are having genetic dissimilarity. So the child which is a heterozygote for many characters of biological parents, you can see whether the child behaves in a similar way. If the biological parent are a schizophrenic, sorry, you can see if after adoption change in environment can bring down the occurrence of schizophrenia, or can it be environmental or genetics. Family studies, I gave the example of schizophrenia. You can relate what is the risk factor. If say the parents have high, both the parents are detected with cancer, then the child will have high chances of detecting the cancer. So family studies also explain the gene and environmental function. If both the parents have cancer and the child does not get any cancer, that means additive to the genetic effect, environmental also plays an important method. Another types of analysis of this understanding genetic and environmental effects is by knowing interaction of single genes, whether what type of SNP or single nucleotide polymorphism is present a particular character. if one detoxifying enzyme is present and that enzyme is overly protecting or not at all protecting, what is the mutation it is haved over the population can be linked to genetic and environmental interaction. When we take obesity, there are a lot of genes which are interacting. Like for example, here gene is leptin gene, LEPR is leptin related, I mean receptor related gene, and then the other POMC is a hormone precursor related gene which are important for synthesis of melanotropin, lipotropin and endotrophins. The deficiency of POMC causes adrenal insufficiency and then obesity. These genes are directly related to MCR4, which are expressed in hypothalamus, that is beta melanocyte stimulating hormone 4 receptor. So whether this gene is the interaction of genetic predisposition for obesity, and then because leptins are very important, they are important because they are there is a pathway called leptin melanocortin pathway which is affecting in determining whether satiety or non-satiety conditions. So you can determine whether single gene by observing the type of mutations or SNP affecting or polygenic genes affecting the conditions, that also, and with the environmental effect gives a clear picture of what kind of gene and environmental interaction is there. By understanding gene and environment interaction, disease can be prevented by reducing exposure to certain environmental risks. The study of gene and environmental interaction is important for improving accuracy and precision in the assessment of both genetic and environmental influence. It's very important in the implications for public health. Evolution also plays an important role in determining the interaction. So other few examples in humans are MC1R, that is again alpha melanocyte stimulating factor, which is present in skin, that is on exposure to UV light. If the person has any genetic defect, then the person has a risk of skin cancer in higher people with who are very fair skin. CCR5 gene is a chemokine receptor type 5 or CD19. It is present on the surface of WBC cells. If there is a deletion, that is delta 2 deletion, if the such person is exposed with HIV, then there is a lower rates of HIV infection and disease progression. PPARG2 gene is related with peroxisome proliferation activated receptor gamma. If you have a mutation from proline at the 12th site to alanine, then the person is exposed to dietary fat intake, there is stronger intake and the obesity can be related. Another gene example is ADRP2, that is adrenoreceptor, that is beta adrenergic receptor. If there is a mutation from arginine to glycine, then if they are homozygotes, then have a greater response to airway with asthma drugs. So these are the references. So on the whole, gene and environment has greater interaction. I started the presentation with explaining what is discontinuous and continuous trait, and then followed by the genetic environmental interaction. We started with the basic of what is a genotype, what is an environment, and then different models of genetic interaction and the significance of this genetic interaction with respect to disease in the reference. Thank you.